Psych 3, Lecture 5, The Visual System. Overview of visual system, physical stimulus. Delta, in energy from external or internal environment, receptor, sensory, transduction, change of neurons' membrane potential, CNS, central nervous system, stimulus, electromagnetic energy visible to humans, that is light, receptor, photoreceptor cells in the retina, central nervous system, CNS, occipital lobe. Light coming into the eye reaches the retina. In the retina, light energy initiates neural activity. At the center of the retina, the fovea is the region of sharpest vision and has the densest distribution of photoreceptors specialized for color. Retina, sclera, cornea, iris, pupil, lens, fovea, optic disc, blind spot, blood vessels, optic nerve. As light enters the eye, it is bent first by the cornea, travels through the pupil, and is then bent again by the lens. The curvature of the cornea is fixed, whereas small muscles adjust the curvature of the lens to focus nearby or far away. The cornea and lens of the eye, like the lens of a camera, focus light rays to project a backward inverted image on a light receptive surface. Inverted image on a light receptive surface. Lenses. Structure of the retina. Retina, light-sensitive surface at the back of the eye consisting of photoreceptor cells and other neurons. Translates light into action potentials, discriminates wavelengths, colors. Fovea, region of the retina that is specialized for high acuity, generates our central vision. Blind spot, region of the retina, optic disc, where optic nerve and blood vessels exit. This area has no photoreceptors. Why don't we see it? Cornea, pupil, lens, iris, retina, fovea, optic nerve, fovea, optic nerve. Cellular structure of the retina. Retina, light, optic nerve. Light activates photoreceptors, rods and cones, ganglion cells. Ganglion cells send signals from retina to the rest of the brain via thalamus. Light, axons of ganglion cells, ganglion cell, cone rod. Photoreceptors, light, fovea, light, retina. Cones are concentrated in the fovea, which includes only cones, not very sensitive to light, but tuned to specific frequencies. Rods are much more numerous, but only in periphery of retina, which includes a few cones too, very light sensitive. Cones are responsive to bright light. They are responsible for color vision and our ability to see fine detail. Cone, synaptic terminal, inner segment, outer segment, cone rod. Rods are more numerous than cones and are more sensitive to dim light. They are mainly used for night vision. Color vision. There are 3 different opsins, photosensitive proteins, that are present in cones. Each cone has only one type of opsin, which is most responsive to a certain color of light, that is, range of light frequencies, small, medium, and large wavelengths. The trichromatic theory states that our perception of color depends on the activation of these 3 types of opsins in different combinations. When M and L overlap too much, red-green color blindness, Peak sensitivity, S cone 419, rod 496, M cone 531, L cone 559. Maximum response percentage, wavelength, nm nanometers. From the retina to the brain, neural pathway for vision, lateral view, eye, optic nerve, optic chiasm, optic tract, lateral geniculate body, thalamus, visual cortex, V1, striate cortex, light, eye, cranial nerve 3, controls pupillary constriction, midbrain. Each visual hemisphere receives input from contralateral visual hemifield, optic tract, optic chiasm, optic nerve, nasal retina, temporal retina, eye, visual field, lateral geniculate nucleus, optic radiations, primary visual cortex, region V1, not only the contralateral eye. Retinotopic map in V1 striate cortex. Parieto-occipital sulcus, cuneus gyrus, calcarine sulcus, lingual gyrus. Light in left visual field activates neurons in right V1, for example. Light in left visual field activates neurons in right V1, for example. Light that hits the fovea, that is, the center of the visual field, activates neurons in the most posterior region of V1, superficial. V1 has a map of retinal position, which is retinotopic map. Neighboring V1 neurons receive input signals from neighboring retinal neurons. Fovea, left visual field. Receptive fields. A visual neuron's receptive field, RT, is the area of visual field that a neuron can detect. That is, the neuron's activity changes when light reaches the corresponding photoreceptors in the retina. As we advance the visual pathway, higher-order visual neurons combine receptive fields and their preferences get more complex. At peak complexity of visual processing, neurons respond to specific classes of objects, like cat, airplane, book, shoes, etc. The receptive fields of many retinal ganglion cells combine to form the receptive field of a single LGN cell. The receptive fields of many LGN cells combine to form the receptive field of a single V1 cell. What drives spiking activity in V1 neurons? The electrode is recording activity in a single neuron in V1. The recording electrode is connected to a speaker that gives a sound every time there is an action potential. The neuron is responding to a high contrast elongated stimulus. Hubel and Weasel experiments, 1959. Electrode system, receptive field, fixation point, light stimulus. Monkey's field of view, fixation point, stimulus, receptive field, stimulus onset, time. Orientation tuning of V1 neurons. Experimental setup, light bar stimulus projected on screen, recording from visual cortex. Record, stimulus orientation, stimulus presented. Discovery of the what versus where pathways. Unger, Leiter, and Mishkin, 1982. Accuracy, parietal lesion, temporal lesion, object task, spatial task, area removed, temporal lobe, object discrimination. Area removed, parietal lobe, landmark, discrimination. Higher level visual processing, two parallel processing streams or pathways. What? Object recognition, occipital, inferotemporal, ventral stream. Where? Spatial localization, occipital, parietal, dorsal stream. Or how? How to act with reference to an object. V1, V2, V3, V4, parietal cortex, MT, V5, inferotemporal cortex, central sulcus. Where what? Where what? V1, WW Norton. Achinotopsia, medical temporal MT area. People with achinotopsia cannot see motion well, caused by damage to MT area, dorsal stream. MT area is at intersection between parietal, occipital, and temporal lobes, still more dorsal than inferotemporal cortex. MT area is at intersection between parietal, occipital, and temporal lobes, still more dorsal than inferotemporal cortex. The inferior temporal IT area object recognition recordings from a single neuron in monkey IT inferior temporal, while these objects were in the neurons RT. Spikes per second, AB. Visual agnosia. Lesions in subject DF, location of LOC in neurologically intact subjects, P smaller than 10 to the minus fifth. Explicit matching task, action task. Patient DF has damage in parts of the ventral stream. She cannot recognize objects but knows how to act on them. Visual agnosia for objects. Failure to recognize objects in visual domain which cannot be attributed to a deficit in primary sensory processing. That is, can see but can't recognize. Recognition can be made with other modalities such as audition or smell or through other sources of information, e.g. actions. Picture of a clarinet. Visual agnosia number 1, 1.2. Face area in IT. No eyes, brush, no color, bunch of degrees. Fusiform face area, FFA, in inferior temporal lobe, response to images of faces in humans. Fusiform face areas, fMRI, when viewing images of faces. Special case of faces, prosopagnosia. Prosopagnosia. Oliver Sacks, the man who mistook his wife for a hat and other clinical tales. Best-selling author of Awakenings. Object agnosia. Object agnosia. Inability to recognize objects includes visual agnosia. Inability to recognize objects visually. Prosopagnosia. Inability to recognize faces or see faces. Face pareidolia. Face pareidolia. Prosopagnosia, face blindness, inability to identify or recognize faces, but no general deficit in object recognition, can occur as a result of bilateral temporal lobe damage, fusiform face area far into the what pathway, but also congenital, can recognize people based on other traits such as voice, gait, or other body features, can often see emotions from facial expressions. Summary of visual pathways in brain. Central sulcus, parietal cortex, MT, V5, V1, V2, V3, V4. Inferotemporal cortex, what, where, HCER 46 FEF MSTI MT V1 PB MP.